fluids, and that is your intracellular and extracellular fluid. Then we talked about the blood and its formed components, plasma. We have the plasma and its formed components, erythrocytes, leucocytes. We talked about the erythrocytes, that erythrocytes are the major component of the blood. If we talked about the RBCs, content of RBCs, then it is 5 to 5.5 million per cubic millimeter of blood. We talked about the lifespan of the RBCs, that is 120 days. And one more thing we have done, that spleen is known as the graveyard of the RBCs. Then we talked about the leukocytes and their further divisions. Leukocytes are divided into three categories. That is your eosinophils, neutrophils, and basophils. Leukocytes basically divided into two categories, granulocytes and agranulocyte. Granulocyte further divided into three divisions, that is neutrophils, basophils, and eosinophils. And agranulocytes are divided into two categories, that is monocytes and lymphocytes. We talked about the eosinophils, that are the part of granulocytes. Granulocytes are the cells that take a particular dye, is known as granulocytes. And agranulocytes do not take any dye. Granulocytes, as the name suggests, granules are present which help to take that dye and become colorful. And agranulocytes do not contain any granule, and that's why they do not take any dye. We had talked granulocytes divisions, that is eosinophils, basophils, and neutrophils. If we talk about neutrophils, the neutrophils take both type of dyes, acidic as well as basic. We talk about the nucleus of the neutrophils. The nucleus of the neutrophils are polymorphic nucleus. You can say, or you can say multi-lobed. It has multi-lobed. Then basophils. Before going to the basophils, percentage of the neutrophils in out of WBCs are 60 to 65%. 60 to 65% all the cells are neutrophils. And coming next to the basophils, basophils are, if we talked about the quantity, number, percentage, it is 0.5 to 1%. Discover very less cells, 0.5 to 1%. Basophils, as name suggested, these take basic dye. Cells which take basic dye from WBC are remarked as basophils. Then eosinophils. Eosinophils that contain, you can say, that take eosin. It is an acidic dye which they take. It covers 2 to 3% of the WBC. You can see in the slide structure of eosinophils, basophils, and neutrophils. Monocytes are also there, T lymphocytes and B lymphocytes. This is all the diagrammatic representation of granulocytes and agranulocytes. Look, eosinophil. Eosinophil, it is orange color dye. Eosin is orange color dye, that's why it is represented as orange color. The nucleus of the eosinophils is bilobed, it looks like a headphone. Then coming next to the basophils, basophil which contain which take basic dye. The next is your neutrophils, neutrophil having polymorphic nucleus, many lobed. If we talked about agranulocytes, then they do not take any dye. First of all, they do not take any dye. They are divided into two categories, monocytes and lymphocytes. The percentage of the monocyte is 6 to 8% of the WBC, and lymphocytes are 20 to 25%, which form our immune system. Lymphocytes further divided into two categories, T lymphocyte and B lymphocyte, which we will do in next uh, +2 chapter about immune system. Basophils is basically secretes three: histamine, serotonin, and heparin that are involved in inflammatory reactions of the body, allergic reactions or inflammatory reactions, in which heparin is kind of anticoagulant, and histamine help in dilation of blood vessels, or you can say it is a vasodilator. In respiration, histamine secrete from mast cell which help in bronchi constriction, or you can say which help in constriction of bronchi. Next is if eosinophil resist infection and allergic reactions. B and T lymphocytes are responsible for immune response of the body. Next we're going toward next formed element, that is thrombocytes. If we talked about platelets and thrombocytes, both thrombocytes and platelets are similar in function, but platelets present in mammals and thrombocytes present all the vertebrates except mammals, because mammals have platelets. This is the basic difference between platelets and thrombocytes. Platelets are present in mammals and thrombocytes are present in all the vertebrates except mammals. Now question is, what is thrombocytes? Thrombocytes are nothing but these are cytoplasmic fragments of megakaryocytes of red bone marrow. These are the fragments. They are the fragments of megakaryocytes of red bone marrow, nothing more than that. These are basically without nucleus, but if you see the smear of platelets, you can see a dense cytoplasmic granule, or you can say basic cytoplasmic granule at the center, which looks like a nucleus, but that is not nucleus, that is only a cytoplasmic granule, dense cytoplasmic granule. If we talk about the size of the platelets, then it is very small in size. This is 2 to 3 micro, and the number of the platelet count, if we talk, then it is 2.5 to 3.5 into 10 raised to power 5 per cubic millimeter. These are very less in number as compared to erythrocytes, or you can say RBCs. Next is lifespan. What is the lifespan? Lifespan of platelets is 7 to 10 days. After 7 to 10 days, they dies off. Two types of diseases are there because of platelets. If platelet count is decreases, then it is known as thrombocytopenia. If the number of platelets count, the number of platelets decreases, then it is termed as thrombocytopenia. And in another way, or in second way, we can, there is another disease, that is thrombocytosis, in which number of platelet is increases. When the number of platelets increases, then it is known as thrombocytosis. If the number is decreases, then it is known as thrombocytopenia. As we know, platelets help in blood clotting. How do they clot our blood? When our platelets get injured, then a substance releases from that platelets, that is your thromboplastin, which is called platelet factor. Platelet also release serotonin, which is a vasodilator. This is most important thing that have to remember. When platelets get injured, enzyme they release an enzyme that is called thromboplastin, which is known as platelet factor. Platelet also release serotonin as your basophils secretes, that act as a vasodilator. So platelet and thrombocytes are similar, but the major difference of the platelet and the thrombocyte is platelets are without nucleus, but thrombocytes having nucleus, and the shape is spindle shape. In case of your platelet is circular, you can say, but your thrombocytes are spindle in shape. Next, we will talk about the blood groups. As you know, all the human beings are different from each other. You can say it is look wise, you can say it is behavior wise, you can say it is blood group wise. There is something different in all the human beings or all the individuals. If we talk about the blood, there is two main types of grouping of blood, ABO and Rh factor. ABO blood grouping based on the presence of or absence of two surface antigens, RBC, antigen A and antigen B. All human beings having blood group A, B, AB and O. All having, not all having, basically A and B antigens are present and plasma of an individual contain two antibodies which are produced in response to antigens. Next slide. Look at this slide. If a person having blood group A, then the antigen on the RBC is A. A question arises here, what is antigen? Antigen is nothing but a kind of protein which are present in RBCs. The protein, this can be monosaccharides, I mean to say carbohydrates. Okay. Blood group A having antigen on RBC is A and antibody plasma is anti-B. It means blood group A having antigen A and antibody B. The donor of who can donate blood to A is obviously group A and O. Why O is there? Because they do not contain any antigen. As we see in B, antigen is B and antibody is A and donor is B and O. In case of O, there is no antigens present in RBCs. That's why it is a universal donor. Okay. Next, coming toward AB. If blood group is AB, then antigen present in RBC is A and B, but there is no antibodies present in plasma. This is, you have to remember that blood group A having no antibodies in the plasma. That's why it can accept blood from AB, A, B and O because there is no antibodies. If antibodies is present, for example, in blood group A, there is antibodies present. For B, if donor's blood is of B, then antibodies kill that. Antibody B is present in blood group A. They kill that. Next, coming toward the next, blood group O. In blood group O, there is no antigens on RBC. but having antibodies A and B, and only O can donate blood to, no other, because there is antibodies present for A and B also. If we take blood from blood group O, take blood from A, there is antibody A, which kill all the antigens or RBCs. They coagulate, they form a clump. If we take blood from B, then there is antibody B, which also kill RBCs and form a clump. That's why O only took blood from O, only from O. So that is all about today. In next video, we will talk about RH factor, RH positive and negative. I hope this video is very helpful to you. Thank you.